So we see interesting things with this with CSF.
Good afternoon, everybody. I'm Tammy Kendall from the Department of Surgery. For those of you virtually attending, please do send us a message if you're not able to hear us appropriately, but we're going to go ahead and get started with our citywide grand rounds. It is really my pleasure to introduce Dr. Darleen Sandoval, who's a professor in the Department of Pediatrics and Medicine at the University of Colorado. I met Darleen in 2007 when I was a PhD student at the University of Cincinnati, where Darleen was a faculty member in endocrinology. And we were at the Genome Research Institute, which turned into the obesity,
Center for Diabetes and Obesity.
He's changed names. In 3 years, I was there like 3 times. But really a great group of physiologists and neuroscientists studying gut hormones, the gut-brain axis, and bariatric surgery. And I can say for sure that my relationship with that group and Dr. Seeley and being inspired by Dr. Sandoval and the work they were doing has totally shaped my career as a bariatric surgeon and scientist. So I followed her career as she went from Cincinnati to the University of Michigan and now to the University of Colorado. Dr. Sandoval is funded by the NIH and serves on NIH study sections. She studies hypoglycemia after bariatric surgery, a very relevant and troublesome problem that we see clinically. And I was really excited to learn that she shares the same passion for me for advocacy for women in leadership in our unique fields as scientists and clinician scientists. So she's to be commended for that work. So I'm sure you will not be disappointed today by her talk on bariatric surgery, an integrative physiologic approach to obesity treatment.
Thanks so much for that introduction, Tammy. It's so fun to be able to reconnect. And I think I'm going to stand. All the audience is over here, so I think I'll stand over here.
You make it stop because of the microphone for the people.
So she turned all of them on, and she said there are microphones above. So if you guys can't hear me again, maybe speak up in the chat. Yeah. All right. Yes, so thanks so much for the introduction, Tammy. It is really fun to be able to reconnect. And it's great to be here, although you can, for those of you in person, you can see based on my shoes that despite the fact that I spent 15 years in the Midwest, I forgot about cute little Mary Janes are probably not appropriate for November in the Midwest. So my research has really always been focused on understanding or being interested in brain control and metabolism. And our latest sort of avenue of research has been trying to look at the gut-brain axis and its role in regulating metabolism. And we do that with 2 different themes. One of them focused on GLP-1 physiology and pharmacology, so gut peptide, that is also made in the pancreas and the brain. And as most of you are aware, that system is being very successfully targeted for the treatment of obesity and diabetes right now. And then the other is focused on bariatric surgery. And we started this work when we were in Cincinnati. And the goal then was let's find that gene that regulates the successful responses to bariatric surgery, and we can find an alternative approach. And I don't know how many, probably like between myself and my collaborators, 25 single knockout genes that we've studied, and we still don't really have that single gene. And so I retitled my talk an integrative physiology approach to obesity treatment, because that's really, I think, what we have seen with bariatric surgery. So no surprise to, I do have one little consultant disclosure to make, but none of that impacts the data I'm going to show you guys today. So I think most of you are all aware that obesity is really an integrative physiology disease, right? So it affects all organ systems, and I'm just showing some of my favorites here. So of course you get an increase in body fat and inflammation. There's impairments or changes in glucose tolerance, and for many people, eventual onset of type 2 diabetes. So the liver, the pancreas, and the muscle is impacted. There's lipid accumulation in the liver. Gut peptides, depending on which literature you look at, can be impacted by obesity, and the microbiome, if you're into that sort of thing, is also impacted by obesity, of course, cardiovascular disease. And then there is an impact of obesity on feeding and energy expenditure and cognition. But we also believe that obesity is basically a brain disease. And so that some of that impaired neurocircuitry that you see with obesity is actually contributing to the onset of obesity. So this slide is actually very outdated nowadays, but we do see with obesity treatment that diet and exercise can be effective. I think it's important to note that this 5% weight loss you can see, 5 to 10% weight loss depending on the intervention that you can see with lifestyle intervention has an important metabolic effect on these patients, right? And so it's not to be ignored. But certainly when you add pharmacotherapy on top of that, you can get some additivity. And again, I mentioned that this is outdated because some of our drugs on the market now or that are going to be on the market are seeing closer to 20% weight loss, so nearing bariatric surgery success. But for the longest time, bariatric surgery seems to be the most effective and sustainable impact on average on body weight. And so that success is really kind of what spurred our interest in trying to take this to the animal model and try to understand why it works. And there are many flavors of bariatric surgery. Roux-en-Y gastric bypass for a long time was the most popular in this particular surgery. You're getting a gastric pouch. You have, I guess the laser doesn't really work, right? So you get a gastric pouch, the midgut is moved up and sutured to that gastric pouch. And so basically ingested nutrients are bypassing 95% of the stomach and the upper GI tract. And then another type of surgery is vertical sleeve gastrectomy. And in this particular surgery, 80% of the stomach along the greater curvature is removed. And this has actually become the most widely utilized bariatric surgery in the U.S. And I'd like to say that we saw this coming, and that's why we started with this model when we were working on establishing preclinical models of bariatric surgery. But of course, like probably the reason why it shifted in popularity from Roux-en-Y to BSG, a part of the reason we started with it is just much easier to do in the mice, right? But it was a very interesting surgery to start with because it turned out that a lot of the physiology was very similar between sleeve gastrectomy and Roux-en-Y, despite the fact that they're very different surgeries. Of course, there are some differences, but there are a lot of similarities. So our typical mouse model of vertical sleeve gastrectomy, our experimental paradigm that we study these animals on, is that we're putting them on a 60% high-fat diet. So we're putting them on a hammer diet, right? Definitely getting these guys obese. We've done variations of that, and we see similar results. But lately, we've been using more hammer diet. And then after 8 to 12 weeks, we do body composition, and we divide them into fat matched groups. And typically that's going to involve a sham surgery and then, of course, the vertical sleeve gastrectomy. Now, we have a brief post-operative recovery period, usually 3 to 4 days. And then, hopefully this isn't or at least Tammy wants to make sure this isn't happening in her patients, but we're putting our animals right back on high-fat diet. So the idea here is that we're trying to understand those effects of surgery that are independent of any changes in diet, and so that is to some extent a limitation of what we see, but it's also extraordinary that we still see the wide array of metabolic effects that we see. So I'm just summarizing a whole list of data. These are some of the very consistent findings that we have with sleeve gastrectomy, and that is that there's a decrease in fat mass. The animals tend to have a preservation of lean mass. The males, if any group is going to lose lean mass, it would be the males. The females are very protective of their lean mass after bariatric surgery, which I think is very interesting. Very apparent changes in glucose homeostasis in the animal models. What we see is a very early improvement in hepatic insulin sensitivity, and then with weight loss, you get the improvement in skeletal muscle insulin sensitivity. And right away, you see changes in insulin, so decreases in fasting insulin and increases in peak insulin. So it goes up really quick, but it also comes down really quick as well. The animals readily change their feeding behavior. So they take in smaller, more frequent meals, and they also, if you give them a choice, so if you give them a choice between a dish that has fat, carbohydrate, and protein, before surgery, they want the fat, and after surgery, they want the carbohydrate. You can also, so whether this is a macronutrient issue or a caloric density issue, we're not sure about, because you can do the same thing with Ensure, which is a sort of a complete nutritional liquid meal. So you can put a full bottle of Ensure on the cage and a diluted bottle of Ensure on the cage, and before they'll choose the full full strength Ensure, but after surgery they want the diluted Ensure. So it's kind of an interesting thing that they're gating their caloric exposure potentially. There are widespread changes in postprandial gut peptides. There are also changes in the microbiome. Whether those changes in the microbiome, again, are just associated or actually causative in any of the success we see with surgery, I think is still unclear. And then we also see changes in lipid handling and bile acid handling at the level of the liver and intestine. And I'm going to talk a little bit about the bile acid handling today, amongst other sets of data. So one of the things that, as we were kind of exploring the physiology of these surgeries, was that the rate of nutrient entry into the system is very rapid. And you would predict this with Roux-en-Y gastric bypass, right? Because you essentially just have a pouch. You don't really have a stomach. You don't have a pylorus anymore. And indeed, this is like, 5 minutes after a barium plus Ensure Gavot, the nutrients are well into the intestine with Roux-en-Y gastric bypass. Now, at the time that we did this study, we were very naive GI physiologists, and we thought that with VSG, it should look more like the sham, because there's still a pylorus there, the stomach is just smaller. But instead, we saw that these animals have, again, 100% of their gastric contents. I think I have that graph here. So 100% of their gastric contents are emptied within 5 minutes. So in both circumstances, we're having this very rapid nutrient entry into the system. And then, so we were very curious about this and wondered if this had any role to play in the success of surgery. And so we poked at seeing whether we could regulate the gastric emptying rate in these animals. And we first of all found that this wasn't just a liquid meal issue. It wasn't just a Gavot issue. If you had them, if you trained rats to eat solid food that had a little bit of radioisotope on the pellet, and you trained them to eat that amount every day, then you could see that the solid emptying was also very rapid in those animals. And that was, we studied that specifically in sleeve gastrectomy. It didn't seem that when you give higher dose, higher caloric density foods, gastric emptying rate will slow down. And we didn't see that with these guys. So if we did a dose response of calories, we didn't see a slowdown. We also didn't see a slowdown with atropine or exendin 4, although a caveat to that is that I have some data in mice, some recent data in mice, where we gave a very high hammer dose of exendin 4, and that peak glucose was lowered. And so it's possible that is more specific to rats. So something we need to evaluate further. But it seemed to us, at least with this track data, that the gastric emptying was really unregulated in these animals. So the other thing that was really key for us in terms of trying to understand, it was a key finding that led us to some data that I'm going to show you today, is that this rapid nutrient entry rate did not seem to be the key factor that drove the increase in postprandial GLP-1. So GLP-1 goes up tenfold after bariatric surgery, after a meal. And if we took animals and we gave the same slow rate of infusion into the intestine of both a sham and a VSG animal, the VSG animals still had, a couple-fold higher, fourfold higher increases in active GLP-1 levels. And so at the time it was kind of perplexing because we were, okay, so what's going on here? If it's not the rapid nutrient entry, what is it? And so the hypothesis that we developed was that maybe there's something about this chronic rapid nutrient entry into the intestine that is causing an intestinal adaptation leading to the increase in GLP-1. And so some fun facts about intestinal physiology. Your small intestine is longer than you are tall. It is the last barrier between the external and internal environment. And so it is a really important organ for immune function, right? Intestinal cells turn over every 2 to 6 days in most adult mammals. It has its own nervous system, of course, enteric nervous system. And its function is not just digestion and absorption. That's what I learned in my physiology class as a graduate student. But it also is important in immunity and secretion of gut peptides that regulate nutrient processing, satiety, and gastric motility. So it is, and in fact, I think I've heard it referred to as the largest endocrine organ in the body, right? So it is an interesting organ to study, and you could imagine that if you're continually having this rapid nutrient entry into the intestine, that it might develop some strategies to try to defend itself against that rapid nutrient entry. And so one of the things that we kind of thought about is maybe the anatomy of the gut is changing, and so one of the things that people do to look at that is they assess the crypt depth, which is where all the stem cells and progenitor cells are located, or the villi length, which is where the majority of the absorptive cells and sort of the endocrine cells, et cetera, secretory cells are located. And so the intestine, depending on how you manipulate it, can manipulate the crypt length and villi length to adapt to, for example, surgery or something like that. And so we measured this in our animal models to see whether perhaps that absorptive surface area is being expanded, and that's how they're handling the nutrients. And so these panels on the right are showing the villi length and the crypt depth, and you can see that. So the sham animals are in black and the VHG animals are in blue, and there's no difference across the 3 sections of the intestine, so the duodenum, jejunum, ileum, or whether we're talking about villi length or crypt depth. So morphology isn't changing. So what else is going on? And so I had a student that was postdoctoral fellow, Ki-Suk Kim, who's now running his own lab at University of Tennessee Health Science Center. And so he was really interested in this question. And so he did some immunohistochemistry and counted the number of GLP-1 positive cells. And lo and behold, he saw about a doubling of GLP-1 positive cells in the jejunum. And so now this may not seem like much, but when you're talking about a very few number of cells to actually see a doubling, we thought was physiologically important. And then up on the upper panel there, the bar graphs, that is showing, so this is just showing that the plasma GLP-1 in either rat or mouse models is elevated. So this is seen clinically, and we definitely see it in our mouse models. The interesting thing about that top panel is that those are actually very low caloric doses. So if you look at the sham animal, they're not going, there is a scale issue on the y-axis, but they're not going up a huge amount. And yet with both sleep gastrectomy and Roux-en-Y, you see a big increase. And we can see that as well. So the bottom panel shows the mouse model, and again, we see that increase. Yeah.
How fast after you do the surgery do you see this increase in cells?
Yeah, so we haven't done anything extraordinarily early after surgery, but in patients, I think it's within a week, I think 10 days. It's very rapid. So I think those early increases in GLP-1 are definitely something about the nutrient entry. But it's maintained over time, and so I think some of that is this adaptation that occurs in the intestine. All right. It's not just GLP-1 that goes up. There are other gut peptides as well, and also this is shown with immunohistochemistry as well. On the left here we have our PYY cells, and this is immunohistochemistry and just counting the cells. And in this case, it's the duodenum and ileum that have that increase. So there's these GLP-1 and PYY are thought to be secreted by the same enteroendocrine cell, although that's sort of the dogma about how this all happens, but it turns out there are distinct enteroendocrine cells, and so we think that's tapping into some of that development. So anyways, we see this increase in PYY cells, and of course we see the parallel increase in PYY, and that's in our mouse model. So we're getting this increase in these gut peptides. We're getting an increase in these cell numbers. And so how is that happening? And so when we were at University of Michigan, we had a conversation. So Linda Samuelson studies intestinal cell differentiation. And this is something I borrowed from one of her review articles. And so basically, as a stem cell differentiates into the variety of types of intestinal cells that get produced, it goes through a couple of different signaling pathways in order to differentiate itself. And so if you're going to be an absorptive cell, then you're going to be exposed or susceptible to Notch signaling. So Notch signaling drives enterocyte development. So these are your cells that are absorbing your nutrients. But there's a whole bunch of secretory cells as well, and they go through ATO1 signaling in order to become a secretory cell. And there are many types of secretory cells. So you've got your Paneth cell, your goblet cell, your Tuft cell, and then of course, for our interest, your enteroendocrine cells. And so I'm showing you this because what we did was try to understand so we did basically some RNA-seq to try to understand which of these pathways might be impacted. So what we did to do this is we took an animal model that had a green fluorescent protein on one of its stem cells. So this is LGR5 specifically that's being labeled here. So we have our animal model that has a GFP on a stem cell, and then we took the animals, put them on a high-fat diet, and did sham or VSG surgery on them. And then after about a month, we took the intestine and we sorted for that green fluorescent protein, and then did our seq. This is work done with Praveen Sethapi at Cornell University, who has a big interest in intestinal cell differentiation and uses these sort of omics approach to try to answer those questions. So just here on the left is just reminding you about the Notch1 versus the ATO1 signaling pathways. And what we saw was that the secretory pathway specifically had significant increases in target genes for that pathway. And some of these, like extraordinarily 200-fold increases. And then, of course, increased gene expression across multiple different gut peptides and precursors for the gut peptide cells. And then at the same time, the absorptive cell lineage wasn't really impacted. I don't think I have that data here, right? So we think that the intestinal differentiation is driving this increase in GLP-1 cell number and is one of the components that's driving that increase in plasma GLP-1 that we see persistently after bariatric surgery. So of course, in science, you want to keep digging, right? So we wanted to try to understand what might be driving, what are the signals from bariatric surgery that might be driving that increase in differentiation. And so we needed to turn to a different tool to do that. And so what we did was we went to intestinal organoids. And so this is really fun stuff. So this is, you basically take those crypts, you isolate them from the intestine. That's where all your stem cells are located, right? And then you put them in a culture dish and you have just the right milieu. And what will develop is this little intestinal organoid that has all the same cell types as your intestinal organ. And then you can use it as a screening tool, right? And so what we wanted to do is try to understand to use it as a functional assay. What compounds can we put on those organoids to increase GLP-1 positive cells. And so the question is, where do we start? Right? There are lots of things impacted by bariatric surgery, so where should we start? So right now I'm going to take you on a little bit of a diverge from this path to tell you about bile acids and their change with bariatric surgery. So these are mice that have had sham or VSG surgery, and on the x-axis are basically a whole cocktail, like all these different varieties of bile acids. And you can see that almost across the board, there is an increase in bile acids. So there's just a couple of species where you may not see the increase, but pretty much across the board, bile acids are increased. Importantly, if we look at, if we do sort of, so here we have a pair-fed control group. So in this pair-fed control group, what they have a sham surgery, but they are pair-fed the amount of food that VSG animals eat. And when you do this, you can mimic the weight loss that is seen with sleeve gastrectomy. And so because of this, we think that what VSG does is modulate food intake rather than energy expenditure to drive that weight loss. But importantly, so these guys, these pair-fed guys weigh the same as the sleeve gastrectomy guys, but they don't have that same increase in bile acids. And so the moral of the story is this increase in bile acids is a weight loss independent effect. Okay. All right. So again, we're always interested in why things happen, and so we were trying to figure out why we get this increase in circulating bile acids. And so it turns out that bile acids are their appearance in the plasma is very tightly regulated and goes through a whole process. And so so if you have bile acids in the intestine, they get transported through ASBT into the intestine. Then they move into the circulation, into the portal vein, where they get taken up by the liver again via specific transporters. Sort of a reuptake process, okay? And so we basically looked at the different components of this enterohepatic circulation to try to see if we could figure out where, what part of the system was modulated such that we get this increase in circulating bile acids. So the first thing we saw is that there is an increase in ASBT staining in the ileum, suggesting that there is an increase in resorption of bile acids into the intestine. So it's definitely getting into the intestine. What about on the side of the liver? Well, interestingly, on the liver side, there's decreased gene expression for several of these bile acid transporters, suggesting that the reuptake into the liver is downregulated. And so if you have this increase in resorption, but a decrease in reuptake, then you could imagine that could be a way that you would get this increase in circulation. And so what we did to try to answer this from a physiological perspective, rather than just sort of looking at expression of proteins and genes, what we did is we gave a stable isotope labeled bile acid, so TCA, and we infused it into the duodenum at a constant rate. And then we sacked the animals a couple hours later and looked to see where that stable isotope ended up. And so the idea, so basically we're going to see because we've given it long enough, we should see the label. If it goes through the whole system, we should see the label appearing in the liver, right? The timing will allow us to see it. And so here is what we see in the plasma. So the VSG animals are in red, and you can see that their appearance of the stable isotope into the plasma is much higher compared to the sham surgery animal. So this is suggesting that it's being resorbed from the intestine and being put into the circulation. Interestingly, we didn't see much of a difference in the liver. It's definitely tending to be a little bit lower. So it suggests that at least even though we're having this decrease in expression of these reuptake peptides, it's enough to get a normal level of bile acids into the liver. So I think it's sort of protecting the liver, that decrease of expression. So definitely that seems like we're getting this increased resorption, that's increasing the levels in the plasma. And that is is important because bile acids don't just serve as detergents, but they're also signaling molecules, and they signal at many different target organs. And so when Karen Ryan was starting her lab at University of Cincinnati, she was really interested in the potential impact of FXR, and so she did some experiments to try to address whether FXR might be important in regulating the response to surgery. So again, I'm showing you this. So potentially why FXR is interesting is that, so it's expressed in the liver and adipose tissue. It plays a role in regulating gluconeogenesis and lipogenesis, and it regulates energy expenditure via a G-coupled protein receptor in the brown adipose tissue. So those bile acids getting into the circulation can act at multiple different organs. And actually, I don't have it here. Oh no, I do on the next one. But it's also potential they're acting on the brain, right? So when you activate FXR in the intestine, you increase the circulating hormone FGF15 if you're a mouse and FGF19 if you're a human. And that can also signal on distant organs to regulate metabolism and feeding behavior. And then, of course, TGR5 is also located in the intestine, where it's thought to be important for GLP-1 secretion. So it is also a potential target here, and I think I'll mention that in a little bit. But let me just show you. This is older data, but let me just show you because I think it really kind of set us up on the journey that we're in as to why we think bile acids are important. So I mentioned that we've studied whole hosts of genes that are important, or that we thought might be important for bariatric surgery. This is the only one that seemed to indicate that it could be important. But again, as I showed you, FXR signaling is important in many different tissues, right? And so it to me represents kind of an integrative physiology gene. But when you look at the whole body knockout for FXR, so here's our wild type animal on the left in white and the black hatched. They lose this. So this is just after 11 weeks of surgery. They lose at least 10 grams of body weight. That is predominantly body fat. But the knockout animals don't lose any body weight at all. Now, it is important to know, excuse me, to note that those sham surgery animals are a little lighter, but that's not a flow effect. Excuse me. And then, like you would predict, since they didn't lose any body weight, they also didn't improve their glucose tolerance. So it's probably the most solid link we have for any potential genetic factor that might be driving surgery. But it really points to bile acid signaling as being a potential important aspect. So we come back to our organoid model. So again, we have this body of literature that supports FXR signaling might be important, that maybe bile acids are important in driving that. And so that's where we decided to start, was with the bile acids. So what he did is he grew his little organoids in a dish. This is staining for GLP-1 cells. So the little green dot represents GLP-1 cells, and these are under basal conditions. And then interestingly, he saw that there was an increase in GLP-1 positive cells, whether he was using primary bile acids or secondary bile acids or taurine conjugated bile acids. So it seemed like the whole cocktail was really driving this increase in GLP-1 positive cells, which is really interesting because, and I've really tried to push him to try to explore this a little further, but all these different bile acids have different affinities for the variety of nuclear transcription factors. So it's kind of an interesting question that begs to be answered for sure. So a question is whether we see this potential increase in GLP-1 positive cells with bile acids. Are we getting this intestinal differentiation because of FXR TGR5 signaling? And so when he did that RNA-seq data that I told you about, he definitely saw an increase in FXR expression in those stem cells. He wasn't able to detect TGR5 in those stem cells. So it really kind of led us to this idea that maybe FXR activation might be playing a role here. And so what he did, this is just a different mouse model. Here we're using a pre-proglucagon, which is the gene that makes GLP-1. Cree mouse crossed to a TD tomato reporter. So now the GLP-1 positive cells are being represented with red dots. And so here's your enteroid with cholic acid. And he had a nice dose response with cholic acid. So those higher doses, you see the increase in pre-proglucagon positive cells. He also gave an FXR antagonist by itself and with the cholic acid. And so what you would predict is that cholic acid with the FXR antagonist should have blocked that effect. And in fact, that's what he saw. So the FXR antagonist alone had no effect on the number of pre-proglucagon positive cells, but when you give the antagonist, you block that cholic acid increase. And so we think that this is supportive of bile acids driving through FXR this increase in enteroendocrine cell differentiation, and that this could also contribute to the increase in GLP-1 that we see with bariatric surgery. But there are several open questions to this. So what time is this over at? Okay, good. I panicked a little. There are a lot of open questions to this still, right? So I think one of the big things is enteroendocrine cell differentiation really a mechanism for the success of surgery, or is it just sort of this adaptive response of the intestine? And can we, if it is just an adaptive response, can we use it to learn more about intestinal function and apply it to intestinal disorder diseases? Is it specific to enteroendocrine cells, or are other secretory cells impacted? We are digging into this question a little bit deeper with some better staining, but our preliminary assessment with NUC2 staining suggests that at least it's not goblet cells that are going up. So the Panna cells proved to be a little bit more difficult to stain. So one of our collaborators is doing that for us. But it suggests that it's not happening to goblet cells for some reason. And then the other thing that has come up is what other aspects of intestinal physiology might be altered. And so that has led us to another set of studies, and this is preliminary data that I'm going to finish with. But basically, so what we've done is we've taken the animals, again, so what we did this time is we have a chow-fed group, a sham high-fat group, and a vertical sleeve gastrectomy group. And we did single-cell RNA-seq on the intestinal crypt and also the villi. And so here is the clustering of all those cells. And you see all the nice pretty colors representing the different cell types. But one of the things that came up intellectually as we were going through this data is that there are actually multiple scenarios that could happen here, right? So you can imagine that obesity has an impact on the intestinal cells, right, and their gene expression. But sleeve gastrectomy, and sleeve gastrectomy, when you do that, could correct some of that. But it could also have its own independent effects that are just offsetting what you see with bariatric surgery. And that's where the data really seem to lead us. And so this overlap in those genes that, obesity is either increasing or decreasing expression. VSG could certainly be reversing that, and there certainly are a subset of animals that we see that with, and so we're calling that a rescue. But there are also specific changes induced by obesity and specific changes induced by vertical sleeve gastrectomy. So that's sort of what these heat maps are indicating. So in the crypts, you're not going to see any absorptive cells. And so we have a proportion of genes that are being rescued by vertical. In this case, we're talking about high-fat diet decreased it. On the right, we're talking high-fat diet increased those gene expressions. And VSG caused the opposite effect. And so, so this was the crypt. The crypt, especially when you're talking about the sort of high-fat diet increasing, it had less of a response. But when we look at the villi, we see a little bit more of a response, especially in those enterocytes. And so again, we have some genes that are rescued, but then we have some genes that are specifically impacted by vertical sleeve gastrectomy. And what has been cool, again, this is very preliminary data, but what has been really cool about this data is that when we look across all these different clusters of cells, one of the pathways that's coming up across all these different cell types is oxidative phosphorylation and myogenesis target genes. Now, these guys, the progenitor cells, don't have mitochondrial activity, and so that's why we didn't see anything there. But certainly we see it in the Panus cells and the stem cells. And it's interesting that VSG is, and I think it's actually this blue is that VSG is actually decreasing it. I actually have a PhD in exercise science, and I've always hung out with mitochondriacs. So I was really excited to have an excuse to look at the mitochondria. And so we've started poking around in the literature, and there is definitely a group of folks that are studying the metabolic responses of various intestinal cell types. And I realize you may not be able to read all of this, but all the different cell types have different, they lean one way or the other, whether they're going to be more glycolytic or more oxidative phosphorylation. And there is certainly, when you're talking about inflammatory bowel disease, there are people that are really trying to capitalize on mitochondrial activity being important in some of these inflammatory bowel disease. The problem I can see with my initial sort of perusing with this literature and talking to Ariana Nathiz, who's at Colorado, is that a lot of this data is based on gene expression and not necessarily mitochondrial activity. And so what we've been doing is trying to actually do a functional assay of mitochondrial activity. It's going to be complicated because, especially when we're talking about the enteroendocrine cells, there's so few cells that to really be able to isolate them and then do the mitochondrial might be complicated. Mitochondrial assays might be complicated, but it's something that we're thinking about and we're working on. And certainly, since we see this increase across the board, across the cell types, we're going to at least start there and just look at intestinal cells generally and see if we can see any changes in mitochondrial activity. So that is something, stay tuned, that we're working on. And we think we have success. The postdoc that I have working on it did her graduate work on hepatic mitochondrial activity, and so I think this particular intestinal inflammatory disease field don't really have metabolism experts that they're working on to do these assays, and so I think we've had some initial success, so we're optimistic. So again, open question is trying to find that functional marker for that increased mitochondrial function. We've also thought about potentially just doing a proteome that we can do on the intestinal, so sort of a mitochondria focused proteome analysis that we can definitely do at a single cell level. And then trying to understand, again, what is the role of that increased in mitochondrial function, if that's what we're seeing. And then, of course, is that FXR bile acid dependent? Is it something else? Is it something more about the nutrient flux that's happening through the intestine, which makes a lot of sense. And so with that, I will close and thank the people that actually do the work around here. So Key has done most of the intestinal differentiation work. This group, this was our fall picture from last year, and we've already had some transitioning. So I've got Kelly Fuller and Tyler Cook added to the group as postdoctoral fellows, and Jenna Nault has been added as a technician. Megan and Sylvania are both moved into faculty positions, but still at University of Colorado in the lab. And then I need to thank Praveen Sethathi, but probably even more so his graduate student, Kieran Laskowski, who really has done the bulk of that, the seq work that we've been working on in the lab. And then just lastly, I'll put in a plug for the group that, some of the advocacy work we're doing for women. And so this is our website if you have any interest in membership, just seeing what we do, or donating. And I would love to take any questions.
All right, so while we wait for questions to come in, as a developing gut microbiome groupie, when you guys are doing your animal surgeries, are you giving a perioperative antibiotic?
We are.
And then
Not only that, but we're flushing the cavity with antibiotic.
So, when you look at your organoid model, how do you account for not having that gut microbial community in your findings, or do you think it's even relevant to not have it?
I'm not a microbiome groupie.
I did it carefully, groupie, not a microbiologist.
It really is. I think that it's possible the microbiome is important in terms of processing those primary to secondary bile acids. I don't know whether it's playing any role in this differentiation process. What I really what we need to do, I tried to get Key to do, and maybe he'll do that now that he's on his own faculty position, or maybe we'll have to just do the experiment in the lab, is we need to see whether intestinal differentiation, it's difficult because to really look at that differentiation, well, actually what we can just do is stain for GLP-1 and PYY in the FXR knockout animal, right? Even if we do it in the intestinal FGF15 knockout, I think it could still be helpful. And just address whether we're seeing an increase in those cells or not. And that would help account for some of that, for sure.
Have you guys done studies in any tissue-specific FXR?
Yeah, we have. And it's like, I actually took those slides out because it is really complicated. So Rohit Kohli's done the SHIP knockout. So what he does is he does a viral hepatic SHIP knockout, and those animals lose weight as you would predict. I think they improve their glucose tolerance just fine as well, but they still had markers of inflammation, and they even have had some sort of exacerbation of inflammation. The intestinal FGF15 knockout has been done, and that is also incredibly convoluted, because what happens there is those animals lose more weight because they're losing lean mass and bone mass. And so it's interesting when we do these tissue specific, we get such a convoluted story. And so there was a paper published maybe 2 years ago that made the argument that it's not just liver FXR signaling or intestinal FXR signaling that matters, it's the integration of the two. And so that's kind of what way that I talk myself into the fact that FXR is still important. It's just because we have this interaction. It's just really hard. How do you study that, right?
We're gonna develop a tissue-specific inducible FXR knockout of the heart, which I think will have less of the phenotype issues that you're seeing with the intrahepatic circulation. But I often think, as a groupie, it would be great to repeat some of these studies in a germ-free model. But the problem is the gut microbiome is integrated within bile acid signaling, and you're not going to know.
Yeah. So you're in a germ-free mouse, the bile acid population
It's going to look totally different. Yeah.
Yeah, sure.
I have a question. So you said the muscle was protective. How do you think? What's because when we do weight loss, then just to diet without exercise, I mean, you lose both.
Yeah.
So
I guess I assumed that for a mouse there is less weight bearing activity to drive increases in skeletal muscle mass. And my understanding, and you can correct me if I'm wrong, but my understanding is that some of that loss in lean mass that happens with weight loss is just there's less weight to carry around. In a mouse that's in a little tiny cage, there's going to be less of that issue. I could be way oversimplifying that, but that was kind of what I always thought about. So yeah, I don't know, and maybe it's something about the mouse model specifically that maybe they just hold on to their lean mass more than a human does.
Does RXR also contribute?
Potentially, because my recollection is, so we detected RXR in the stem cell and vitamin D receptor in the stem cells.
Yeah, I mean, and that interaction is good for you.
Yeah,
It can be a nightmare.
yeah, sure.
So if you just gave an agonist to a fat mouse, can you
It's so the FXR system is very complicated, right?
I know that
Yeah.
sounds so simple, but what agonist and how much?
Yeah,
I know,
exactly.
But have you tried it?
We have not tried it. I want to say that there have been some there are data out there on it, and but I think that there is a bunch of side effects that go along with that.
And I think that the literature supports maybe glucose regulation more than it does food intake, energy expenditure effects, if I remember the literature.
So it's actually FGF15. If you give it to the brain, definitely suppresses feeding. So I think for FGF15, there's definitely a satiety role to play. But yeah, it's true, right? Like it's maybe not necessarily an overall body weight regulator, but could it be playing a role in NAFLD or type 2 diabetes potentially?
So as someone who's not a groupie in any of these areas,
I like groupie. If you're an expert, that doesn't make you.
All those things are scary, and I'm glad no one heard that.
I heard.
Temporally, from the start of surgery, when do you see each one of these, and can you use that temporal change as a way to really drive that mechanism at each step?
Yeah. So are you talking about just sort of the metabolic responses?
You pick the response, because each one could be different, and the metabolic response is, I think, the first one, right? I mean, you see that in 5 days. So how fast do you see changes? And if you could actually do that, then you can use your organoids to do the metabolism stress.
Yeah. Yeah, so we in terms of intestinal differentiation, we're not sure. We've only done the chronic stuff, right? Sort of waited a month or 6 weeks and to look
You
at
could
counting
what if
the
you
cells,
double
but-
those cells
Yeah.
the day of the surgery? And
Yeah, it's a great question. Like, how fast? Yeah.
and as a postdoc, you would like that experiment because it's way better than waiting a month.
For sure. Yeah.
And the organoids may be a good way to start to go after some of the metabolic parameters. I'm very old school with metabolism, and I struggle with more glycolytic and more oxidative because I grew up in a generation where the only time cells used the mitochondria was when there was a need, a demand for something, and it was pretty much off unless there was an event. Cells live fine by glycolytic metabolism.
Yeah.
And this new wave of mitochondriacs think of it very differently than that.
Yeah.
I mean, I think that way because the evidence is pretty good that once you start inducing mutations in the mitochondrial DNA, apoptosis happens right away for those cells. So that was a really nice explanation for that. But those organoids could be really a good way of going at those questions.
Yeah, I think so too. Yeah.
Have you ever done this in rat?
Yeah. Yeah. So we haven't, so I should say, we've done the surgery in rats. So the gastric emptying rates, that data was all rats. We have not done to see whether the number of GLP-1 positive cells are elevated or not. I speculate that the GLP-1 is definitely elevated at very low caloric loads. So I speculate it is.
We talked in the afternoon too, so you have been using Exendin 4. Any thoughts on using the newer ones? Because they're way a lot more, it seems like very different than what Exendin 4.
So you're speaking to the data. So we have data in mice where we have data in rats where we gave Exendin 4 and didn't really see a slowing of gastric emptying rate when we did that. We haven't. I'm sure we have the glucose levels from that particular study, so I'd have to go back and look at it. Because if you're going to measure acetaminophen, you might as well get a glucose level. But what we did recently was a study where we gave Exendin 4 to mice after sleep gastrectomy, and the peak glucose was substantially reduced. So when I see a reduction in peak glucose, I speculate that gastric emptying rate has been slowed. There could be other things going on as well, but I speculate that that is one component of why it's so suppressed. We don't have acetaminophen in those guys, so I'm not sure whether it is or not. So a little bit of a disconnect, and I'm not sure if it's just sensitivity to Exendin 4 or what's going on. I would expect, liraglutide or semaglutide to have that same flattening of the glucose curve that Exendin 4 has. They're all for glucose regulation. They're either suppressing gastric emptying rate, some believe, or they are, stimulating insulin secretion. And so, we have not done the studies where we look to see whether, and actually, I know you're looking at this. There is also, and I should connect you with her. There is a peds endocrinologist. Actually, she's in nutrition, but she's doing some studies with the peds endo group where they are taking post-bariatric surgery patients that aren't losing weight or that are having weight regain and giving them the agonists. To see whether they can induce weight loss. And we have talked about doing the same thing in the animal models so that we can have a little bit more of a back and forth because we can get tissues and stuff like that. But we haven't done that yet.
We give it, and it works. Again, most of them have been a year or two out, but lately I've been seeing some that are within that window. I have no data, which I asked you. When do you start GLP-1? We are now, at 6 months. Obviously, they are not losing weight because I think most of the weight loss is majority is coming from GLP-1 related mechanism. Then maybe giving it, it has always worked for me, but most of them were really out. What I was getting at, so Exendin 4 is probably very different than the newer ones, such as Zepatide. That is just blowing everything out of the water.
Right.
Even compared to semaglutide, I think semaglutide will probably only be used if you don't get tirzepatide.
So except the oral formulation.
Oral formulation doesn't work. It's very low dose. It doesn't.
I thought it was having extraordinary success.
Oral formulation?
Yeah.
Semaglutide? No. It's equivalent to only 0.5 milligrams. The dose now is 2.4 milligrams of semaglutide. 0.5 is not very effective. It is effective, but so we don't use actually, or maybe you guys use, but it's almost not at all.
So I will say that I don't believe that bariatric surgery mechanism is GLP-1.
Okay.
So that could be some of the reason why you have activity, right? It's because, especially when you're talking about and in the animal models, this suggests that our data are, we have a animal model where we can reactivate glucagon in a tissue-specific fashion, and when you put it back in the intestine, you definitely see the normal plasma GLP-1 response that you would predict. like knocking out GLP-1 receptors, like, or GLP-1 secretion, like for multiple models that have done this, it could be a human versus mouse thing, but those models all lose weight, but, just fine. And so it's like,
I mean, I'm not me, but one.
yeah. So I think, I think flat GLP-1 plays a role in acute regulation of feeding and acute regulation of glucose control. Whether it has a role to play for chronic, I'm not sure.
It's fascinating. The more you dig, I think the story you told is fantastic.
I wanted to wait till the end.
Yeah,
Knock
last
everything
question.
off
Yeah.
So there's some reports that the sleep gastrectomy models will really shift the hematopoietic compartment, really shift some of these, which is what I'm really excited about.
Yeah.
Do you see that in your model? Have you looked at that? And
Yeah, and so I apologize because that is definitely outside my wheelhouse, but we do. So Durga Seger looked at it a little bit in our animal model, so she's really interested in sex differences with weight loss, and so we sort of compared our weight loss models. But she's a I don't know, what would you call it? So she crosses obesity and immunology, right? And so and then with Orman McDougall, we also looked at the hematopoiesis in those guys, and there were definitely some differences. But I'm not sure we really understood what was going on. He definitely sees this interesting decrease in bone adipose tissue. Yeah. And so I think it's there. It's interesting. It's outside my wheelhouse again, so I'm not really sure what it means, but I think it's very interesting. And these surgeries, a lot of these patients have decreases in bone mass, right? So there's definitely something integrative going on there.
Well, I'm curious, just you have this real change in this kind of like liver filtration system, and you're getting a lot of these bile acids sort of getting into circulation, and that's just gonna change so many of those balances. But he's shown that change in fat mass in the marrow seems, at least when I talked to him, that seemed to be the sort of the major driver he was thinking was causing the change.
Yeah.
Your data suggests maybe it's both and.
Yeah, sure. Yeah.
And do you see a change in? I mean, if you looked at immune cells on the intestine, sort of in the?
No, we haven't.
I'd be really interested to see if it's changing that sort of myeloid population.
Absolutely, absolutely.
That could have huge impacts.
Yeah. That is something we have done. If you're interested, I can send you some.
So what happens if you were to sleeve gastrectomy in a normal weight animal?
Actually, they do just fine. They will lose some adipose tissue. They'll lose some weight, but
I mean, the physiologic changes, do they? I mean, you mentioned that the obesity may be a different kind of milieu.
I think we haven't looked incredibly in depth at like the things that we've looked at in terms of differentiation or anything like that. We've mostly, often we're doing it just to like, we have a new surgeon and so they're practicing and they'll do it on some lean animals and they lose the mass. They'll even improve their glucose tolerance a little bit. They have that same sort of peak in glucose and rapid return with baseline that you see with the high fat animals. We've also done the version of like switching them to chow. So do the HGN, switch them to chow, and there's additivity in the amount of weight loss and improvements in glucose homeostasis that you see in the males. And the females, it's more complicated. It's a little more wishy-washy. But yeah.
Yeah, right. All right. Thank you so much, Darleen. I think we'll wrap up the session here.
Yeah.
Thank you for your time.